VSL test interpretation, the total gram-negative count, brought to you by the Cornell University Milk Quality Improvement Program and the Cornell Dairy Extension Group. In this module, we'll discuss why the total gram-negative count is important for your milk quality, what gram-negative bacteria are, how the counting is done, and how to interpret the results of this test. Gram-negative bacteria, unlike spore-forming bacteria, are easily killed off by the heat of pasteurization. This means that they must have entered the milk after the pasteurization step when we find them in pasteurized milk. We call this recontamination or post-pasteurization contamination. Many types of gram-negative bacteria can multiply quickly to very high levels, even at refrigeration temperatures. This results in standard plate counts above the PMO limit of 20,000 cells per milliliter, sometimes within the first week of refrigerated shelf life. We often see sensory defects formed by these fast-growing gram-negative bacteria. Gram-negative bacteria are named after their reaction in the gram stain test, and they are everywhere in the natural world. They live in soil, in bodies of water like lakes and rivers, on plants, and in the bodies of animals and people. Gram-negative bacteria enter raw milk on the farm in several ways, including through mastitis and the contamination of milk with cow feces, but fortunately these bacteria will be killed by pasteurization if the milk is properly collected and handled. Unfortunately, the dairy plant environment can reintroduce gram-negative bacteria to the milk through contaminated water, equipment, or workers. Gram-negative bacteria multiply in refrigerated milk, causing spoilage early in shelf life. We notice this spoilage as off flavors like bitterness, off odors like rancidity, curdling of the milk, or occasionally because the milk changes in color or consistency. In the video to the right, milk has become ropey due to the gram-negative bacterium Rhinella. The type of gram-negative bacterium we find most often in spoiled milk is called Pseudomonas, and there are many different species of Pseudomonas that can spoil milk. Post-pasteurization contamination with gram-negative bacteria can be reduced through regularly scheduled preventive maintenance of equipment and by encouraging employees to follow important good manufacturing practices such as hand washing. When you send us your milk samples, we use a machine called a spiral plater to spread some of the milk onto a petri dish of crystal violet tetrazolium agar, and we then put this petri dish into a 70° Fahrenheit incubator to allow the bacteria to grow. Crystal violet tetrazolium agar, CVTA for short, usually allows only gram-negative bacteria to grow. After 48 hours in the incubator, we count the number of colonies of gram-negative bacteria that have turned red on the petri dish and report this to you as the number of colony-forming units per milliliter. There's a column for the total gram-negative count on your VSL report, and it's reported for each test day of refrigerated shelf life. If we haven't detected gram-negative bacteria in a milk sample, then the count is reported as below the detection limit, less than 20 E. The E stands for estimated. If the count is above the detection limit, then it is highlighted in red. Samples with high gram-negative bacterial counts often have low sensory scores, as is seen here for sample 52 on day 14 of shelf life. On this day, sample 52 has 760 million gram-negative bacteria per milliliter of milk. Its sensory score is 1.3 out of 10, and the sensory panelists reported that the milk was coagulated and bitter tasting. On the initial day of testing, we also perform a rapid test to predict whether low levels of gram-negative bacteria are present in a milk sample. A positive result on the rapid PPC stress test means that the sample has an increased chance of spoiling due to gram-negative bacteria. Thank you for watching this module. If you're interested in taking classes with us, learning more about our research, or participating in our voluntary shelf life program, please refer to the information on this slide.